Well, thanks, John, for the invitation. It's great to come to Fort Collins. I came a little early and got to sit through some of the course, which was fantastic for me. And this is really a great opportunity to tell you about some of our most recent work. We've been working with switchgrass for 3 years now. And what I'll tell you about is really what we've been learning over these last 3 years. And I'll tell you a little bit about a set of projects that we're setting up. So it's a great time for me to get feedback on how we're focusing some of our projects, trying to look at local adaptation to climate in Panicum virgatum switchgrass. So as John told you, my background's really in ecology and evolutionary biology. And my lab is generally interested in understanding the process of local adaptation. So how the environment might impose selection on plant traits that lead to populations evolving to have high performance in a local situation. So I really don't have a background in breeding, but I have a pretty strong background in quantitative genetics and trying to understand response to selection. So ultimately, I think actually the kinds of questions my group works on are very similar to many of the projects that the community of breeders for drought are interested in. And so I'm interested in the same sorts of traits. I'm interested in how drought and water deficit can impose selection on suites of drought adaptation traits, like drought escape, avoidance, and tolerance. And I have a really long-standing interest in how plastic traits might evolve. And I think it's really important to realize that many of the traits that we think of as being escape, avoidance, or tolerance characteristics might be inducible or plastic. So that's a theme that you might see come out throughout the talk. So the type of questions my lab tends to focus on, we'd love to discover genes that are responsible for novel adaptations in natural populations, especially related to stress tolerance and the abiotic environment. We'd like to know more about the types of genetic changes, be they structural or regulatory, that might be important for the evolution of tolerance. We'd like to know more about the role of plasticity in that local adaptation. And if we can actually get to those genes and sequence them in a population context, we'd love to try to link effects of particular alleles with some understanding of their evolutionary history. So are they rare? Are they common? Did they arise once and spread? Those sorts of questions. And most of my career has actually been working with John and Jim Richards and focusing on Arabidopsis thaliana, so our model plant, our model organism, where we have really great genomic resources and actually fantastic collections of natural accessions from around the world. And so in this context, we've been looking at natural variation in whole organism physiology and drought tolerance. And we've completed a lot of quantitative genetic studies looking at water use efficiency as measured using carbon isotope methods. My background is really in QTL mapping and association mapping, so we've been completing those types of studies in Arabidopsis, including some efforts to clone QTL. And then we've also been very interested in completing studies of drought-induced gene expression using a wide diversity of natural accessions and asking whether drought-tolerant or drought-susceptible lines might have different patterns of gene expression induction under stress. And then most recently, we've been trying to take advantage, like everybody, of the new sequencing technologies to understand natural variation in populations. And so, like I said, a lot of this work has taken place in collaboration with John and Jim, but I'd like to add that really my background didn't train me for physiology, and both John and Jim, I think, have really been mentors, teaching me an enormous amount about plant-water relations, physiology, and drought. And then I'd like to mention that a postdoc who's been really a leader in all of our work with Arabidopsis, Dave DeMarais, is actually in the audience, and he's going to tell you a lot about our Arabidopsis work tomorrow morning. So Arabidopsis has been a fantastic system, and we're going to continue working with Arabidopsis for a long time. But there are some limitations to working with Arabidopsis. It's small, and as Jim can tell you, it's hard to collect a lot of whole plant physiological measures on this plant. It's hard to impose really realistic stress. We don't know much about its ecology in the field. It's very weedy, has a very short lifespan. And these things have always limited, I think, some of our work. We've also not taken the work in the field And as an ecologist, I've always wanted to actually place these sorts of systems in the field. So John's doing that now. I think in 8 days, John's taking off to Sweden, where he'll spend some time studying Arabidopsis in the field. So we've been looking for systems that would be native to North America, where we can ask very similar questions, but actually incorporate field work. And so we started thinking about a variety of different grass systems and have ended up working with panicoid grasses, C4 perennials. And today I'm going to tell you about our work with Panicum virgatum switchgrass. So switchgrass is interesting for a number of reasons. It's native across North America, has a very widespread distribution all the way north into Canada, throughout all of the eastern United States, throughout the Midwest, throughout most of the Southwest, and then actually also occurs down through Mexico and Central America. And it occurs in many different habitat types, in forests, in riparian areas, in tallgrass prairie, where it can often be a dominant, throughout the Southwest, in dry, arid conditions, and even on coastal areas and basically coastal marshes and prairies. So there are two really well-characterized ecotypes of switchgrass. There's a lowland type and an upland type, and there's some spatial patterning of the occurrences of these types. So the upland types tend to occur in the north and are quite often freezing tolerant and also thought to be more drought tolerant. And the lowland types tend to occur primarily in the south. They're much more vigorous than the upland types, but tend to be freezing susceptible. And then there's a sort of transition zone where we see lots of variability in these ecotypes. Along with that is ploidy variation. So there are tetraploids and octoploids. We find out that most lowlands are actually tetraploids, and most upland ecotypes are octoploids, although that's not strict. There's great resources. USDA GRIN has hundreds and hundreds of samples across North America. There's a long history of agronomic work, initially for improving switchgrass for forage, and more recently for interest in biofuels. It's very productive, even productive on marginal lands. It can be very high yielding, from 7 to 10 tons per acre. And then for me, what's been really exciting is, given the growing interest in biofuels, there are a number of resources that are being generated to have genomic tools for breeding and improvement. So the Department of Energy and the Joint Genome Institute have been spending lots of resources to generate ESTs and back libraries and genome sequence and linkage maps. And these are all the tools that we basically use in Arabidopsis to study natural variation. So here's just a quick picture of upland and lowland types to give you a sense for these things. Here's a classic lowland type. They tend to be a bluish color. They have thicker leaves, wider leaves, fewer tillers, tend to be much more vigorous. Here's an upland type. They have thinner leaves, darker green leaves, many more tillers, shorter, much more, excuse me, less vigorous in general. But please interrupt me if you have any questions along the way. So, yeah.
Real quick, is the higher ploidy tending to happen with more northern types of species that have to adapt more to abiotic or mostly cold stuff? I'm just noticing this all the time.
In general, I think people describe that phenomena. I'm not so sure that there's great data that I've never seen a study that really argues that well. Certainly in virgatum, I think that this seems to be true. For me, one of the exciting things is it spans this really incredible climate gradient. So across 8 USDA cold hardiness zones and over 60 inches of precipitation. And in Texas alone, we see almost 60 inches of precipitation gradient that Panicum virgatum can handle. And so we'd like to ask, how do these local populations, are these local populations adapted to these conditions? And what can we learn about mechanisms of tolerance by looking at this variation? So we actually have lots of evidence that these populations are locally adapted. In the early 60s, Calvin McMillan, a researcher in Lincoln, Nebraska, and also in Austin, Texas, drove around North America and collected accessions of lots of different dominant grasses, including Panicum virgatum. He was a contemporary of Klaus and Keck and Heisey, and he took advantage of common gardens. So he'd take these clones from around North America, grow them in common garden, and be able to ask how much of the variation did he see was genetic, how much was a plastic response to the local environmental conditions. And then he could look for patterns between that genetic variation and, for example, latitude of origin. And Calvin McMillan's work showed that there's an enormous amount of genetic variation, an enormous amount of gene by environment interaction, and also some pretty strong gradients, in particular with latitude. And he was very interested in flowering time and growing season length and performance in these grasses. And then subsequently, as people became interested in these crops for potential biofuel, there have been a number of traditional agronomic field trials, which I think of as tests for local adaptation in some sense. So researchers, many USDA researchers, including Mike Kassler, have grown population samples of Panicum virgatum across North America and asked how do they perform. And what they find is really widespread gene by environment interaction for performance, which in these home versus away experiments you can interpret as evidence for some local adaptation. So here's just one example of a set of northern and southern accessions grown across a latitude gradient, and you can see that the northern accessions perform well in the north and the southern accessions perform well in the south, for example. There really hasn't been much breeding in Panicum virgatum, and so far really the model is if you want to grow for forage or biofuels, you just take advantage of this notion of local adaptation and you plant cultivars or population samples that came from some local source. And people have talked about the spatial scale of this in terms of plant adaptation zones. Okay. And then, of course, people are interested in this crop as a biofuel feedstock. And there's some evidence that this could be economically and energetically viable. Here's a figure, a nice picture representing the potential yields, a study that came out of Ken Vogel's lab in Lincoln, Nebraska, working on farm scale trials, so growing switchgrass. Here is the input of fuel resources given a certain amount of biomass obtained and some pretty reasonable estimates for conversion rates. You can see you can produce quite a bit of ethanol fuel output. Now, this is under pretty good conditions that, to be honest, I think would compete with agricultural products for food. And so a real important thing to consider is whether we could grow switchgrass on more marginal lands and actually still have viable yields that made it energetically and economically viable as a biofuel. And then sort of compounding that is these marginal lands right now are likely to become more marginal in the future with climate change. And so our project is really centered around asking, is there evidence for local adaptation in switchgrass to climate gradients and at what spatial scale? Which gradients are most important? What are the mechanisms, traits, and genes that are important for current or future adaptation to climate? And if we can, try to say something about how switchgrass might perform as a biofuel crop under future climate environments. And ultimately what we'd like to do, I think we're a long, long way from this. I think we'd all like to do this. We'd love to, based on our knowledge of the local conditions, be they either spatial or temporal conditions, and some knowledge of the plant material, try to predict or forecast what our yields might actually be. So we've been playing around with this using some existing crop modeling systems, including Almanac with Jim Canary, a USDA researcher in Temple, Texas. And we've been parameterizing his Almanac model using one of the dominant cultivars of switchgrass, Alamo, and basically making yield predictions in the future and across space in the context of what we know about soils and water nutrient balance, the climate in those conditions, and what we know about Panicum virgatum, basic physiological characteristics at the stand level. And we've been using some of the existing IPCC climate scenarios to try to project performance in the future under climate change. Again, these are just some really early efforts, but there are some interesting things that pop out to me from these simulation studies that I think could potentially direct our work or also John's trying to trip me up here. Or also breeding efforts. So for example, one of the dominant ideas in terms of breeding switchgrass right now is that we'd like to take the lowland, highly productive cultivars and move them north. They'll have a longer growing season in the north, and they yield very high, but they tend to be freezing tolerant. So this is a perennial. You plant it once, and you might get harvest for 10 or 15 years. If a rare, really cold winter happens and you lose your crop, it's a huge detriment to growing that cultivar in the north. So people have been talking a lot about freezing tolerance. Well, if you look at the climate simulations, it's pretty clear this little dotted line is what we believe to be one of the major sort of breakpoints for freezing tolerance for some of the dominant cultivars. And that line actually moves quite a bit under some of the future climate scenarios. And so it may be possible to move some of those lowland cultivars farther and farther north, just because in 20, 40, 60 years, the climate will actually allow them to live there without any breeding for freezing tolerance. We also see some increases in yield under these models, largely because of increased precipitation in the Midwest. And we also see some pretty severe declines in the Southwest, which are those marginal climates that I think people are really considering switchgrass for as a biofuel crop. So none of this incorporates genetic variation, the potential for adaptation, breeding, or altered management. And so part of our project is to look at genetic variation and see if we can incorporate it into these kinds of models or simulations. Okay, so our approach really is to take field trials in common gardens, the same sort of approaches that have been used in the past, but couple them with plant physiological studies and also some genomic studies to try to understand mechanism. And so we have 11 different field trials planted across 6 states using a combination of northern and southern lines, upland and lowland cultivars, and tetraploids and octoploids. And then we have 3 common garden sites located in Texas where we're doing experimental manipulations using rainout shelters. And I'm mostly going to tell you about our rainout shelter work today. And it's been a huge effort. So we got this grant in 2009, and the first year or two have really been spent in collecting material and generating plant material and building structures to impose our treatments. And so here's just some pictures of the effort to build our irrigation systems and basically trench our plots so that we could put in liners to separate our irrigation applications. Trench warfare going on here. Here's 3 postdocs to dig a ditch. We've been challenged because University of Texas at Austin is not a land grant school. And so we haven't had a lot of infrastructure to help us set this up. And we really, the group of postdocs has been fantastic in actually pulling off setting up these sites. We've also got a lot of advice from a faculty member in the irrigation department at Texas A&M, Guy Phipps, who helped us design some of these experiments. So here's the main experimental design. We have 2 field sites, one at the Lady Bird Johnson Wildflower Center, which is just south of Austin and is on a relatively poor, shallow upland Edwards soil. And then about an hour north, we have the same basic experimental design in Temple, Texas, on a pretty rich blackland prairie soil, a much more agronomic scenario. And then at each of these sites, we have structures that exclude rain, and then irrigation systems that will allow us to apply treatments. And the design centers on 4 blocks, and each block has 6 subplots, and each subplot is receiving a particular irrigation treatment. And then within each of those subplots, we have 10 different genotypes that represent upland and lowland and northern and southern plant material. And they're replicated twice in each subplot using clonal division. Okay, so it's a split plot design. Our treatments are applied at the plot level, and we've been using mixed model analyses to analyze data collected under the sites. These are big, monstrous plants, so we give them 1 meter squared apiece in these designs. So they look like big football fields to me. So we have 2 sites, 10 genotypes, 6 precipitation levels, and 8 reps, so about 1,000 plants total that we're trying to keep track of. And then the idea is to measure a host of traits that we've talked about a lot in the class since I've been here and also in the symposium. We're measuring lots of gas exchange, instantaneous gas exchange on the plants, trying to do a good job collecting plant water status, leaf water potentials. We're really interested in flowering time and growing characteristics. And then sort of my end is really to add a genetics and genomics component. And so we're looking at both transcriptome and metabolomic data. And the ultimate goal would be to try to use some of this data and the variation we see among the accessions to predict biomass performance under stress. And for me, what's really exciting is there's been a huge investment to build these structures, but they're perennial. And so we're going to start applying these treatments, and we may be working with these plants and these plots for 5, 10 years. And so we'll be able to look at sustained drought events, recovery following precipitation, and even long-term acclimatization that might happen under our different experimental plots. I'm also a QTL mapper, and so we would like to actually try to discover genes that underlie some of these differences. And so we also have some experiments to do QTL mapping. These are outbred species, and they're tetraploid, and so it's pretty complicated mapping. We're using one existing mapping population, which is a cross between Alamo and Canlo. These are both lowland cultivars that people have studied quite extensively. And so in this QTL study, we have about 200 full sibs. They're tetraploids, but fortunately, these tetraploids show disomic inheritance. And so basically, it's like mapping in a diploid. There's an existing linkage map that we can use, and we've clonally replicated this, again, under one of these rainout shelters. And in this case, we've actually been using sort of a row planting of spaced plants and a row-based irrigation system. And the idea here will be, hopefully, we can detect QTL within the parents for physiological traits and performance traits, and also try to study gene by environment interactions. And then we're also doing some of these cylinder experiments that allow us to have some really nice control of soil dry downs and have access to roots and a sort of ability to do more fine scale physiological measurements. So again, I'm not trained in agronomy, and so it's been a real challenge to think about what are the treatments we might want to impose. How do you do a good job imposing drought on a perennial like this in the field? We certainly have to take into account sort of the annual amounts that we might apply, the seasonality that we might apply, how the events happen, the length of the dry interval. There are many, many decisions that we've had to make. And this is where I'd love to get people's opinions. What we've ended up doing is using historical records and a stochastic weather generator. And so we have about 100 years of data in Temple, Texas, and 100 years of data in Austin. And we basically have taken the 10 highest years, the 10 lowest years, the 10 that span the 75th and the 25th percentile, the means, and the ambient patterns that we see. And we collected statistical parameters that describe those patterns of precipitation and then use those in the stochastic weather generator to basically make new time series of precipitation events. And then we're lumping those into weekly applications and applying them using basically spray devices on the corners of each of the plots. And then in the context of our QTL studies, we've been using basically drip tape in a row agricultural setting and basically reapplying a little bit under evapotranspiration predictions or manipulations that decrease evapotranspiration a particular percentage. And here's just some pictures after 1 year, in the first year of establishment. So these were built in 2010 and planted in the spring of 2011. And we've had 1 year of growth under well-watered conditions. And you can see they're pretty amazing plants. Here's an example of the Temple site on pretty rich soils. And here's our QTL mapping population. And one of the lead postdocs, David Lowry, who's actually a very tall guy. And you can see by the end of the summer of the first year, he can pretty much hide in our plot of Panicum brigatum. So the first year, we well-watered the plants, and we've started collecting some data to basically describe the material that we're studying. And here's just data looking at productivity and basic growth characteristics. So on the x-axis is just the name of each of our 10 genotypes, and then we're looking at tiller production, overall dry biomass production, height of the plants, and basal area of the plants. And the dark gray bars correspond to basically agronomic cultivars, things that have been collected and maybe manipulated through 1 round of selection, maybe 2 rounds of selection. And then the light gray bars are natural accessions that we've collected from Central Texas and Southern Texas, material that's really not been explored at all in the context of switchgrass biology. And so you can see there's an enormous range in biomass production in that first year, several fold changes in total biomass accumulation, and a lot of differences in sort of growth architecture in terms of tillering, height of the plants, and basal area of the plants. And then likewise, we've been collecting lots of gas exchange data just to learn a little bit about how the plants perform across the season. And here's just plots of days of that first season and instantaneous gas exchange measurements and some fluorescent measurements. And basically what we see is that there's tons of genetic variation in these instantaneous measures of physiology, but they show really strong time by genotype interactions. So the lines, it's a little bit hard to see here, but the lines often peak at a different time period or flatten out at a different time period across the season. And then likewise, if you look at our plant material, there's very, very, very big differences in the time to flowering in the field. So the season length is really going to be variable among our lines with respect to vegetative versus reproductive growth. And that's going to be something we're going to have to incorporate very carefully into our analyses. So here's an example of 2 of the most South Texas lines, the highest yielders in our first year, flowers 60 days later than the rest of our material. So a really important component to keep in mind. So we just started applying our treatments this year in February of 2012. And here's just some soil moisture data collected from these decagon probes spanning February to current times, with this being the low, the 25th, what is that? Mean, ambient, 75th, and the high. And so we can sort of track our soil moisture decline under our various treatments. And then we've started to do our fizz campaigns just in the last month. And so again, we've focused quite a bit on trying to measure in paired ways plant status from leaf water potential, gas exchange data, trying to learn how to keep a bunch of lycors running. There's a couple of ecologists, Christine Hawks and Phil Fay, that are both involved in the project that are very interested in ecosystem processes. And so we've been using whole plant chambers to try to look at stand, gas exchange, and net ecosystem exchange. We've also been looking at soil respiration. And then trying to collect leaf samples that are paired with all of this physiological data. And we're trying to stock away lots of this material. And the material could be used for looking at transcriptome, looking at metabolome, proteomics, hormones, you name it. And so one thing I want to emphasize is that this is this big infrastructure that we're developing, and we're really excited about trying to get people to come collaborate with us. If you have any particular traits that you might be interested in studying in this type of material, let us know and come. Take part in a field campaign. There's tons of plant material to harvest for your particular assay. And so here's just a little summary of some of the first data from one of these field campaigns. So again, and this is at the Temple Shelter. These are really rich, fairly deep soils. Here are the genotypes on the x-axis, and looking at pre-dawn water potentials, midday water potentials, and photosynthesis. And this is sort of just representative of the material. We see lots of differences among the accessions, so lots of genetic variation, pretty strong treatment effects. In this case, we're only looking at 3 treatments, keeping it simple, our first campaign. This is the low, the mean, and the high treatments. And basically, we see large treatment effects, large genetic effects, and some G by E for characters. So for example, if we look at pre-dawn water potentials, AP13, which is basically our wild type line that DOE JGI is sequencing, actually seems to have a pretty different profile than some of the other upland accessions. And likewise, we see that it's really pretty sensitive to the drought that's been imposed so far, with respect to photosynthesis. So we'll be making more field campaigns this summer, and we'll be doing this for several subsequent summers as well. Okay, so we've just harvested those leaf samples, so I can't really tell you about any of the genomics yet. But I will tell you about the technique we're using and some of the pilot experiments that we have completed. So a real important methodology that we've been working on in the lab is a tag-seq based method for looking at gene expression changes. So this is a method that was developed by a postdoc in the lab, Eli Meyer. And the idea is to collect total RNA from these leaf samples, fragment that RNA, turn it into cDNA, and then barcode each of those samples, pool them, and sequencing them using a next-generation sequencing technology. And the real power of this is that the prep is pretty quick and fast and cheap, and that we can pool these samples and cheaply sequence them fairly deeply. And the output of the data, we take the data from these short reads, and we map them against the transcriptome. And in this case, we have a really nice transcriptome that's been developed by the Joint Genome Institute and the Nobel Foundation, again, off of this AP13 genotype. And every time we map one of these short tags, we basically can count it. And then we have count data that should correspond to the occurrence of particular messenger RNA molecules in a sample. And so the idea would be we can count samples under a stress condition. In this case, we have 100 tags. In a non-stress condition, we have 50 tags. And then basically directly invoke the fold change that has occurred due to the treatment. This is slightly different than most of the RNA sequencing that people are doing. Most people are sequencing total RNA. Instead of getting a short 3' tag, they'd have short little reads across the whole transcript. And my argument is that's really not a very relevant thing for us to be doing right now, in the sense that we quite often know very little about alternative splicing or more complex characteristics of that full transcript. And as long as that 3' tag is unique and gives us count data, that's what most of us want to ask when we study differential expression. And so by just using 3' tags, we can really cheaply, fairly deeply sequence and get accurate measures of gene expression. So we've spent about a year and a half trying to work through this methodology, and we've played with some various characteristics. For example, the read length of the tag, and this is just showing that we've been collecting 50 base pair data using the solid sequencing platform. But what if we were to actually save some money and sequence a shorter tag? Do we see really any difference in our estimate of differential expression? You can see, no, it really doesn't seem to matter very much. 50 base pairs, anything over probably 35 base pairs is fairly unique in our transcriptome database. And likewise, we've sequenced some samples very deeply and then have tried rarefaction analyses. And here's just an example of looking at how many genes we detect in a leaf sample versus how many sequencing reads we actually generated for that sample. And you can see that it plateaus off about 5 or 6 million high-quality reads will actually detect almost 80–90% of the transcripts in that sample. Okay? So there's still some real challenges, I think, to this approach. Our biggest one is annotation. So Panicum virgatum is about 30 million years diverged from sorghum, and probably 40 million years between maize and certainly much greater to rice. And so many of our transcripts, we have no annotation. We don't know what they are. They're totally novel things. And then likewise, most of the annotation that does exist is pretty distant. And so I don't know whether to believe it or not, to be honest. And then likewise, we're going to look at genetic diversity. And one question is, how much will sequence polymorphisms in our tags influence our mapping efficiency, for example? And then finally, we're working with polyploid material. And I think it's definitely a question whether or not we can uniquely identify the homologs in the 2 subgenomes of this tetraploid using short reads. So it's not a perfect method, but it's very exciting because I think at the current costs, we now can actually pair this type of data with lots of samples and pair that with lots of physiological measures, and I think make some progress beyond what we've been able to do with microarrays. So right now, it costs us about $10 to prep a sample. And to get about 5 or 6 million reads using the Illumina platform, actually with 1/200 pair-end reads, is about $60. So for $70, we can take a leaf sample and very accurately get counts of transcripts. So we've completed one drought stress experiment as a pilot that I'll walk through. Am I doing OK on time? This was the first experiment we did with switchgrass. And we basically took Alamo AP13 clones. This is our wild type plant that's being sequenced with lots of genomic resources. We clonally replicated it through tiller division. And then we've been growing plants in greenhouse conditions using gallon pots. So we grew plants, controlled for size among those plants, grew them to a nice size so there was lots of leaf material that we could sample, and then withheld water. And we withheld water from these plants for 14 days. And this is just a plot of the water content in those gallon pots over that period. And then we basically added water to field capacity in our controls. And then along the way, we collect some gas exchange data and also measured leaf water potential. And we took them to a pretty severe, pretty low water potential, minus 2.5 there on day 13 and day 14. And then we harvested leaf tissue samples for these RNA-seq studies. And I should say that we harvested samples both pre-dawn and midday. And then on the late morning of the last day, we actually re-watered half of the droughted plants and then let them recover for a while and then also sampled tissue. So this experiment should let us ask questions about drought-induced gene expression pre-dawn, midday, and then a recovery phase. And then here's just some gas exchange data paired with these samplings, and you can see that the stress was significant enough to reduce gas exchange and also impact some fluorescence characteristics of the leaves. And then likewise, our recovery, we actually see plants recover with respect to photosynthesis and some of those, again, fluorescence characters upon rewatering. So I think we have sort of a nice time period. We stress them well enough, but not too far, and we actually can see some recovery in the plant material. And I should say that there are two postdocs that really led collecting this data, Mike Aspinwall, a physiologist, and Dave Lowry, an evolutionary geneticist. And so here's, so we get these samples, we extract RNA, we prep them, and we sequence them. We sequence over 100 samples in this case. And we map these against the AP13 transcriptome, and we end up with count data. And in this case, we analyze the data using a mixed model analysis that deals with the repeated structure of the design. And then we could actually make contrast for the particular questions of interest. So in these samples, we find about 25,000 unique transcripts or unique tags in leaf tissue, which is comparable to what exists on most microarrays that you would see currently being produced. Under the drought stress condition, we fit a model that looks at treatment effects, time of day effects. We sampled both pre-dawn and midday, as well as an interaction. And here's actually the number of differentially expressed genes that we identify in these experiments. So we detected over 10,000 genes that had a main effect of treatment. And about equal numbers of these were upregulated, and about equal numbers of these were downregulated. This is actually not so similar to what we've seen in soil dry down experiments with Arabidopsis thaliana. We see several thousand genes, well, almost a comparable number of genes that show a circadian pattern. So differences between pre-dawn samples and midday samples. And then we see several thousand genes that actually show a treatment by time interaction. So these genes are genes that are differentially expressed in response to the soil drying, depending on whether it's in night conditions, early morning conditions, or midday. And then likewise, we separately analyzed our recovery experiment in the same framework. And in this case, we're comparing stressed, control, and recovering plants at different time points in the day. And we detect over 1,500 genes that seem to respond to the recovery treatment and that show a time effect. And here are just some Venn diagrams sort of describing the overlap of the gene lists that we detected from these analyses. Okay, so this is really just a pilot study to get us started, to proof that this methodology will work. and we want to implement it in the field. But I think we've already learned some pretty interesting things. A large fraction of these genes are unknown genes. They have no names. There seems to be no tag to anything else we've seen in another organism. We do see lots of genes that we recognize from other studies. Lots of genes involved in redox regulation, things you might imagine involved in osmotic adjustment. Certainly aspects of photosynthesis changing, lots of transporters, and then of course some interesting transcription factors that we're really interested in. But I think one of the neatest things from this study is this comparison of patterns of gene expression in response to stress in a diurnal context, pre-dawn versus midday. And so we've only got this data very recently, but I'm very interested in looking at the difference in genes that are up or down regulated under a pre-dawn versus midday condition. So just to convince you that the methodology works well, we also have done a lot of qPCR validation. So here's just an example of, 10 or 15 genes that we picked from our gene lists, where we did sort of classic cyber-green qPCR assays on samples. And you can compare under the day conditions and the night conditions, the stress, the recovers, and controls. Or you can just take all of this data and look at the relationship between our gene expression estimates using qPCR versus this tag-based RNA-seq. And you can see that they're really highly correlated. There are only a few cases where we see some discrepancy between the qPCR and our count data from RNA-seq. And my guess is in those cases, it probably has something to do with homologs, where in the case of the RNA-seq, we're actually lumping together more than one gene, whereas we're separating them out based on qPCR. So again, there's no annotation for these grasses, panicoid grasses, and so we've had to try to generate annotation using some of the existing approaches. So this is a custom MapMan annotation against maize, okay? And this methodology basically gives us some tag of annotation for each of our transcripts that match to maize. And then we can ask whether there's any enrichment of particular categories, biological functions, related to our stressed or recovered treatments. And I would say this is a very early annotation, but it's a nice way to actually look at some global patterns. And so you can see in each of these cases, there's significant enrichment under these different MapMan categories. So we can see that under the stress condition, photosystem-related things are downregulated. We see some downregulation of fatty acid metabolism, upregulation of abscisic acid metabolism, for example. Again, many of these things are things you might anticipate. For me, one of the most striking things is to see transcription factor families that I don't think we've really detected previously in other systems. And so those might be unique responses in these grasses. So this is looking at our wild type line. Now what's going to be really fun is when we can actually look at some of the genetic diversity that we know has a different susceptibility to the drought. So for example, trying to compare the upland versus the lowland accessions and to see whether there are any unique biological processes that function in one or other ecotype is certainly where we're going with this. And then likewise, this is a small data set, and maybe it's too early to try to do this, but in theory, we'll ultimately have thousands of samples that are paired between RNA-seq and metabolism and physiological measures. And certainly, we'd like to build predictive models that try to link mechanistically changes in gene expression to changes in hormone abundances, to changes in metabolites, and ultimately physiological traits that we've measured. So this is only looking at 100 or so points and just asking, do we find strong relationships between any of these transcripts and either the physiological measurements that we've made or metabolites? And one thing that I think is a real exciting aspect of this type of approach right now is there are a lot of statisticians who are thinking hard about this problem. And they've been developing some new approaches to include both linear relationships, but also nonlinear relationships. We've never really been able to search these big data sets for nonlinear relationships. And just last year, there was what probably is the only science paper published based on just a statistical analysis. A paper that came out on this new methodology called the maximum information criterion, or mixed statistic, which is basically an analog to the Pearson product moment correlation, but allows for nonlinearities. And so here's just an analysis looking at how many significant, after a Bonferroni correction, correlations do we see between leaf water potential and transcripts, be they linear, positive, and negative, or nonlinear. And you can see we find on the order of hundreds of transcripts that show some pretty remarkably tight relationships with interesting characteristics like leaf water potential, photosynthesis, stomatal conductance, or some of these fluorescence measurements that we've made. And here's just a pick of 4 examples where you can see some really nice linear relationships and even some nonlinear patterns in the data. Okay, so do I have 2 or 3 more minutes? So we've primarily been working with Panicum virgatum, and it's been a really exciting and interesting system, in part because of the natural populations that exist, but also because of the agronomic interest. But it's also a pretty tough plant to work with. So I told you we need a meter space for plants in the field. In the greenhouse, they take over my greenhouse in a couple of months. They're also obligate outcrossers, and they also have fairly large genomes. And so along with our work with Panicum virgatum, we've been looking for close relatives that might make nice diploid models. And so we've been really interested in a small clade of relatively close species that have small genomes and are diploid. And we've been focusing primarily on Hall's panicum. So Hall's panicum occurs throughout the Southwest. It's a diploid, with 2N being the base number for panicum, of 18 chromosomes. Its genome size is about 550 megabases. It's predominantly selfing. We can cycle it really fast in the greenhouse. It has sort of 2 ecotypes that are, in some ways, correspond to these upland and lowland types that we see for Panicum virgatum. These are varieties Hallii and varieties Philippis that, again, occur across the Southwest. And so basically, we've been going out and collecting accessions of this native grass across the Southwest in a variety of different habitats. We find it in relatively wet East Texas. We find it at high elevation Sky Islands in West Texas. We find it in Chihuahuan Desert. And we've been taking these accessions and crossing them and making traditional genetic mapping populations. We just got funding to generate our core cross between these 2 subspecies into a real population. And then the idea here is to actually take clonal replicates of this perennial diploid and plant them in the field and actually do field experiments at the sites of origins of the parents of the crosses. And this will let us ask about bits of the genome that might underlie local adaptation of these types. Actually in the field. And a lot of the work with Panicum Hallii has been led by, again, a postdoc in the lab, David Lowry. And this is just an example of the 2 different varieties, the Hallii type and the Philippis type, and some of the variation you see in the F2 for biomass and tillering characteristics. And so again, this may be an interesting way for us to actually map genes that might be relevant to climate adaptation and also growth characteristics that might be important for biofuels. And the hope here is that we could learn something, find genes easier in this diploid that might help us inform genes that could be important in the agronomic type. And then sort of along with this, developing these resources and collecting this material, we've been collaborating with DOE Joint Genome Institute to actually sequence this diploid. And the idea here is that although they've spent a lot of money and effort and are perhaps the best genome assemblers in the world, right now they can't assemble the Panicum virgatum AP13 genotype because of the tetraploid nature of the genome, the polyploid nature of the genome. And so by sequencing this close relative diploid, we're hoping that we can actually help them generate scaffolds and align and conduct some comparative analyses with this agronomic species. And so we've been generating short read, Illumina reads, and 454 reads, and then working to assemble those. And at the moment, we have about 91% of the genome in assembly at about 100x coverage. But I would say that this genome is still fragmented into small little pieces. And our strategy is actually to use our linkage map to pull together those scaffolds to a chromosome-based assembly in the coming year. And it's already been useful, this assembly. We can blast it against the existing genome. We can map our RNA-seq tags when we do RNA-seq in the species. We can actually align it to the current assembly of the Panicum virgatum genome as well. Okay, so I'm sorry if I'm running a little bit over, but just conclude by saying I think there's already really good evidence for local adaptation in switchgrass. And I think that functional diversity is going to be really critical for switchgrass improvement. At the moment, the cycle for breeding switchgrass is at least 5 or 6 years before you can phenotype because of the establishment phase of these stands. And so for the breeders, they're really excited about using a genome selection type approach that the students would have learned about in class this week. So there's lots of genetic variation that we've seen already in growth, morphology, physiology, and productivity. We can do experimental manipulations to impose stresses. And when we do that, we're already seeing accumulating effects that result in genotype by environment interactions. And the hope is that we can use some of these new tools to actually look at the mechanisms that relate to these differential responses. And ultimately, I think these genomic and genetic tools will be important for actually understanding these mechanisms, and maybe they could help to actually improve agronomic switchgrass in the future. And so I'd just like to end by saying the project's been a really exciting effort, but largely because there's some really great postdocs that have been working on the project. Eli Meyer was a really key person in the lab who developed a lot of our molecular techniques and also bioinformatics scripts. Mike Aspinwall and Sam Taylor are both ecophysiologists that have taught me an enormous amount about physiology and have also helped to set up these big experimental manipulations and run field campaigns. Jason Bonnet is a field technician who's just been the master coordinator of all of our efforts. And then David Lowery is actually an ecologist and evolutionary biologist that's really pushed forward with this diploid close relative. And so I'd just like to end by mentioning a couple other collaborators. In particular, Christine Hawks, Tim Kitt, and Phil Fay, they're all ecologists working on the project. And then Jim Canary, who is a USDA researcher who does agronomic modeling. And then the real fantastic efforts of folks at DOE JGI, including Dan Roxar and Carrie Berry and Jeremy Schmutz, who have helped us with generating genomic resources. And then NSF, Joint Genome Institute, and TAC for computational resources. Thank you very much.